So now we look at the xylem tissues. Xylem tissues. So xylem tissues basically have two functions. They serve as a support and also transport. But for today, we're going to learn actually for transport. How these xylem tissues involved in the transport. So in our syllabus, we need to know that xylem actually consists of different types of cells. So in the flowering plant, xylem tissue consists of, in our syllabus, basal element, xylem fiber, and xylem parenchyma. Okay, now in this flowering plant, we do have another one called, Trackit, but Trackit actually is not really, it's not in our syllabus. Okay, so you look at the structure of the xylem tissues, you can see that some very large vessel elements, can you see that? These very large cells, right? So this large cell actually termed as the, can you see that? Hollow, empty, forming a tube-like structure. So those are the vessel elements. Okay, those are vessel elements. And you can see that we have the dot. This dot actually is the pore that allow water molecule to move in and out. So this structure is known as the pit. Okay. And then you can see that we have some living cells here. So those living cells, can you see that? Those living cells, they are known as the parenchyma, okay, or xylem parenchyma cells. Okay, so within these structures, we do have these how is it that? Can you see that the pit that connect two cells together, the pit. So this pit is known as pair pit. Clear? Pair pit. And we do have the trackit as well. Okay, trackit with a tapering end. Okay, so in our syllabus, our concerns, our focus will be a vessel element, parenchyma cells. Okay, these two, and also the fiber. Okay, so let us look at xylem vessel element first. When talk about xylem vessel elements, so their involvement in the transport of the water and dissolved mineral ion. Okay, so if you look at these xylem vessel elements, they have many elongated cells arranged end to end. When they become matured, so the end wall actually broken down, forming a continuous tubule. So when this vessel continue to develop, the cell undergo apoptosis or programmed cell death. So means that vessel element eventually they are the dead cells. Okay. So each vessel element begin life as normal plant or lignin actually going to lay down. So when talk about lignin they are very hard. They are very strong substance and impermeable to water. Now this one highlight totally impermeable to water. So as long as the lignin start to build up, more and more lignin build up, so the cell gradually will die, leaving a complete hollow tubule. So means that they don't have nucleus, don't have cytoplasm. So therefore minimize the resistance for the water transportation. Correct? If you want to transport water, you have to make sure that they are hollow. Okay, so therefore higher volume water can flow through the hollow continuous tube per unit time. Okay, now then you can see that in terms of the cells, the lignification, so the lignification, they have two forms. One is the incomplete lignification, another one is extensive lignification. So lignification process actually take place. So when the plants, they are young, when they are young stems or the root, so the lignification pattern they use will be annular, ring, or spiral, means spring. Okay, but when they become older and older, need more support, because tall tree. So when they need more support, then it slowly change to the reticulate or pitted vessel. Okay, reticulated or reticulate vessel or pitted. means that this is more extensive lignification. So what actually happened here is they will choose either one. So when the young stem or root, when they are young, they will choose annular. So annular won't change to spiral. Are you clear? Annular won't change to spiral. So annular will remain annular, spiral will remain spiral. So why annular and spiral? Because when annular and spiral is used, allow the wall to be stretched, the vessel to be stretched, to provide the support for the vessel during the elongation and the growth. So it means that it's not very fixed. You try to imagine that the annular and spiral, they give a flexibility for the vessel elements to elongate, to become longer cells and grow. Extensive means that the cells won't be able to grow because extensively deposited with this strong material called lignin. Okay, so we have talked about annular, spiral, reticulates, and pitted. Let us look at the structure. So this form, can you see that it forms a ring, right? So this one is termed as annular. Okay, termed as annular. So this one is, you can see that something like spring, right? Something like spring going down like this. So this form is termed as the spiral. Okay, spiral. And then you can see that this was something like ladder. So this form is called reticulate. And you can see they got the pits there. So this one is termed as the pitted form of the lignification. Okay, but this one is not, guys, be careful. This one is not vessel element. I will tell you guys why later. Why not vessel element? So it can be a trackit, it can be others. But the thing is, cannot be vessel element because vessel element must not have the wall, the end wall here. Okay. So fill up. So number one, we just now know that is annular. Okay. Number two is a spiral. Number three, we have the reticulate or reticulated, and number four is called pitted. Okay, so number one and number two used for the young plant. Number three and number four only for those when you go taller and matured plants. Okay, so you can look at this diagram and identify some of them. So let us start the identifications. Okay, so you can see that, guys, this one. Can you see that? Spiral form, right? So you can write down this one, spiral. Okay, and you can see this one, something like ladder, right? So this is reticulate. Okay, now talk about this. This was very clear cut ring, so it means that annular. Okay, so this one actually something like a ring but spiral, right? Something like a ring and spiral. So this one actually is a spiral. Okay, so this very clear cut spiral, both of them. Okay, how about this? Okay, so happen we cut, this is actually annular. How to differentiate between annular and spiral? Annular, actually the distance larger. And because the parts without the lignin is quite, okay, can you see that it's not strong enough? You can see that they form a curve. So this annular. So it means that this one is actually annular. Okay, this one is a mixture. Top part is a spiral, bottom part is annular. Okay, because different cells. Okay, and then consider this one, dot, right? So this one actually is the pitted. So in exam, you need to know how to differentiate them. Okay. So next, we do have this structure called pits. A pit is a non-lignified. Okay, pits, they are non-lignified. Okay, they can see it as a gap but it's not open pore. Always remember, pit is not open pore. Okay, so they are crossed by permeable unthickened cellulose cell wall, fully permeable. Okay, so the pit in one cell linked with another neighboring cell allow water molecule to move out laterally. So either to bypass the airlock or to the surrounding living cells. Okay, so let us look at this. What's the difference between the pits and plasmodesmata? Because in the chapter 1, we learned about plasmodesmata, but we didn't learn much about pits. So today, let us look at the differences between the pits and plasmodesmata by having this annotated diagram. So let us look at these pits. Okay, so what's mean pits? If you look at the pits, basically, I can draw two cells. Okay, this is first xylem vessel element, and we have another xylem vessel element. Okay, so initially they are living cells, so they do have very thin primary cell wall. Okay, very thin primary cell wall. Okay, so when they are growing up already, okay, when they grow up already, then you can see that lignin start to deposit. Sorry. Okay, lignin start to deposit, but the deposition of lignin may not be complete. Okay, lignin deposit. Oops, I think draw this one better. Can see that? Okay, so this red color is because of the lignin. Okay, but you can see that some part of the plant cells without the lignin. It's kind of, can you see that? So those arrow parts here means that they are without the lignin. So this blue arrow indicates who? Indicates pits. Are they open pore? No, they are not open pore. Okay, if you label it, so you still have the primary cell wall. Can you see that? And the lignin forms secondary cell wall. Okay, can you see that? So is it open pore? It's not open pore because in between of them, we still have the cellulose cell wall. So this is pit. Now, how about plasmodesmata? If you compare to the plasmodesmata, plasmodesmata is or are open pore. Okay, they are open pore. So means that if I draw this, two cells, generally they have very thin cell wall. They are living cells. Okay, can you see that? And they are perforated with the pore. They are open pore. So this one is termed as plasmodesmata. Can you see that? Okay, so means that they are open pore. No cell wall in between of them. So label this is just a cell wall. or primary cell wall. No secondary cell wall, okay, at the plasmodesmata. So you can draw this into the activities, slot there. Okay, so we have done for this part. I will show the differences between the pits and also the plasmodesmata. So if you look at the diagram here, you can see that some parts that without the lignification. So those are the pits. So the pits allow the water molecule to move out because you cannot move through this way. Can you see that? You cannot move one cell to one cell, right? Why? Because lignification is impermeable to water molecule. So very important to know what are the adaptation of xylem vessel element for water transportation. Okay, so this I say that because we want to have the transportations of the the water molecule, so they must have a certain adaptation. So what is the adaptation? First, lack of cell contact, such as the nucleus and cytoplasm. So no organelle. So no end wall in the individual xylem elements, so means broken down. The wall of the elements are lignified, and they do have the presence of the pit. And last, you can see that the diameter is 0.01 to 0.2 millimeter. So let us look at this, how each of them helps to make the plants or this xylem vessel element functional. Okay. So vessel element, they are started with a normal plant cells. They do have the cytoplasm, they do have the nucleus. Okay, so they are elongated cells. Okay, I draw two of them. Get three of them lah. Okay, they're elongated cell. So they're stacking like this. Okay, with the very thin cell wall. Okay, very thin cell wall. Initially. But when the plant become more and more matured, what will happen here is this area, okay, the end wall, they are broken down. Okay, end wall broken down. Completely broken down. Forming a continuous tube. Can you see that? Forming a continuous tube. This longitudinal sectioning here. And then to strengthen, okay, to make this vessel element strong, okay, because this column will under tension. As I said that when you have something flow inside, so the pressure will change. So the change of pressure will make this xylem vessel elements to collapse. So in order to prevent the collapse, okay, so lignification actually take place. So you can see that the lignin start to deposit. So sometimes you may have the pits there, where the lignin deposition take place. Okay, the red color is lignin. So the parts that without lignification is termed as the pits. Okay, so what are the adaptations? So first of all, look at this. Number one, no cellular content. So what does it mean no cellular content? So it means that no nucleus, no cytoplasm. Basically, they are dead cell. Are you clear? No organelles. What's so important about this? If there are no nucleus, no cytoplasm, no organelles, actually they produce a minimum, or they actually allow, okay, or they give a very minimum resistance for the flow of the water. Are you clear? Sorry. So they provide a very minimum resistance for the flow of the water, okay, in this xylem vessel element. Second, if we look at the end wall here, so no end wall. Okay, when there's no end wall, so it can actually allow the water to form a continuous column. Okay, so this is very important. The transportation of water will be fast because they are forming these continuous columns. Are you clear? Okay, third, if you look at the structure here, I summarize, Third, so lignification. This is very, very important because the lignification actually prevent the collapse of the xylem vessel element. Are you clear? If without this lignification, what will happen? They will collapse. Are you clear? Because of the pressure change. Low pressure, then collapse. Okay, but because for lignification, because it won't burst, it's like our everline burst. So in this case, nothing to do with the bursting. It's more to the collapse. Okay. Now next, number 4. Presence of the pits. So this pit actually allow lateral movement of water molecule. So this is very important to bypass airlock. So means that if in case any airlock in between, they can actually move out. Okay. And number 5, diameter. So diameter is between, now this value you have to remember is between, okay, diameter is between 0.1 millimeter, sorry, 0.01 millimeter to 0.2 millimeter. Okay, now a bit difficult to explain about this. So it's small, okay, small enough to maintain a column, to maintain the water column, but it's not too large, okay, to break the column. Are you clear? It's small enough to maintain the column and also large enough to transport large volume of water. Are you clear? So this range is very nice range. Okay, so this range is not too small. Okay, if very, very small, then we won't be able to transport large volume of water. But it cannot be too large, because too large you won't be able to maintain the water column. So this is a very nice diameter to make this xylem vessel element functional. So we total have 5. So according to the ranking, guys, according to the ranking, number 1, 2, and 3 are the most important. If the question asks you about adaptations, 3 marks, I always go for number 1, number 2, and number 3. If more than 3 marks, then I will go for number 4 and number 5. Okay, so you can take a photo here before we go to our notes. So lack of cell content, such as the nucleus and cytoplasm. What is the good thing about this? Because the total lack of the cell content in the xylem vessel element provide uninterrupted pathway or very minimum resistance for the mass flow of the water molecule. Now we're going to learn about this mass flow. The movement of the water molecule in the xylem is termed as mass flow. It's not the osmosis, because osmosis is too slow. Okay, it's a mass flow. Okay? And it cannot be osmosis because no membranes involved. Okay, so no end wall in individual xylem elements. So again, the lack of the end wall provide an uninterrupted pathway for the mass flow of water, and the water molecule form highlight a continuous column inside the vessel element. Okay, and wall of the vessel elements are lignified. When they are lignified, so lignified cell provides structural support, preventing the vessel from collapsing inwards when the pressure inside the vessel element is low because of the flow. Okay, and presence of the pits. So in the case of the airlock or air bubble, pits in the wall of the vessel allow water molecule to move into and out of them. So pits are also allow the water molecule to move out to the surrounding living cells. Okay. So diameter of lumen is about 0.01 millimeter to 0.2 millimeter. So this range of the diameter is wide enough to have high number of water molecule move out through the xylem. However, this range of diameter is narrow enough to maintain the water column, reducing the tendency of the formations of airlock inside the vessel elements. Okay, so it means that we want to maintain, it's small enough to maintain this diameter. Okay, but it's not too small until we cannot transport the large quantity of water. Okay. So next, for Trackit, I forgot to put it in. Trackit just for information only, not in our syllabus. So Trackit actually is, they are lignified also, they are lignified, but with tapering end. So you can see that you will compare between the vessel element. Vessel elements, no tapering end, completely broken down. Trackit, they have pits in between. So tapering end. This is how you differentiate between the Trackit and also the vessel element. Okay, so it does not have opening end to form vessel, but they do have the pits to allow water molecule to pass through. Their role is to transport the water as well. Tracheids, they are more primitive as compared to the vessel element. So if some woody plants, non-flowering plants such as fern, you may see the tracheit. So tracheit actually is much more smaller in diameter compared to the vessel element, but they are the primitive one. So what's mean primitive? Means that those lower rank plants, they will use the tracheit. So evolution take place because tracheit, the plant become bigger and bigger and larger, tracheids too small to sustain. So therefore evolution take place, then the plant actually develop this vessel element. Okay. So we do have the xylem fiber as well. Xylem fiber also, they are lignified. Okay, but they do not involve in the transport. They actually help to support the plant. Because when the plant grows taller and taller, then a lot of this cell actually turn into the xylem fiber. So this xylem fiber actually, they are lignified, so means that the wall are strong. But they don't involve, they don't involve in the transportations of water, but they involve in the support. So because of lignification, we do know that they are dead cells, have no living content for xylem fiber. Okay, and next we have a last type of the cells called xylem parenchyma. Xylem parenchyma are normal plant cells. Okay, they are normal plant cells. Their role as a storage. So storage means that they do not need to carry out photosynthesis, so they do not have chloroplast compared to other plant like leaves, okay, in the mesophyll cells in the leaf. Okay, so because they do not involve the support, their cellulose wall are unthickened. Okay, so means that they're thin. Basically, parenchyma, when you view under the light microscope, they have very thin cell wall, and they contain all organelle but no chloroplast. Okay, you try to imagine in the xylem, in the stem there. So in the stem, do they need to carry out photosynthesis? No need. They don't need to carry out photosynthesis. So they serve as the storage cells. Okay, so with this, I've done for the xylem vessel elements.